A very good morning to all of you. So today's our topic is that of Willson disease. Willson disease is one of the frequently asked question as a short note. It's important topic for UG as well as for PG both. So Willson disease. What you will write in Willson disease? First thing is that Willson disease is what? It is a it is disorder of which metabolism? It is a disorder of your copper metabolism. Okay. What Willson? It is a disorder of your copper metabolism. Okay. Second thing, Willson. How the name this Willson comes? Okay. So there was a famous British neurologist. There was a famous British neurologist named as S. Willson. Okay. His name was S. Willson, who discovered first this disorder. Okay. First this disorder. Okay, so on the name of discoverer S. Willson, the disease is named as a Willson disease. Alright. And what kind of inheritance pattern? It is a autosomal recessive disorder. Alright. So what is Willson disease? It is what associated with metabolism. It is a disorder of your copper metabolism. And Willson name is given by after the discoverer. He was a British neurologist, that is the S. Willson. He discovered and reported first case of Willson disease. And after the name of this disease is known as Willson disease. And pattern is that of autosomal recessive. What is the etiology of this disorder? So it is due to the mutation in ATP7BG. ATP7BG. Okay. I shared a mnemonic on how you will remember the gene ATP7A and 7B. So you should watch that video. So by this that trick you can easily remember which gene is associated with Willson and which is associated with Menkes. Okay. So in Willson disease, which gene is defective? ATP7B gene is defective in your Willson disease. This question is asked in MCQ and Viva also. That which gene is defective in Willson? So the ATP7B gene is defective in your Willson disease. Alright. Okay. And now next come. Before understanding the thing, you must know what happened normally in the copper metabolism. Okay. What happened normally in the copper metabolism? How copper is metabolized normally in the body. Because when you know the normal things, then you can understand easily what abnormality occur in case of Willson. Okay. So what happened in you normally with the copper metabolism? How copper is metabolized in the body. Okay. So what happened? First of all, what? You are taking copper in the diet. In diet. Okay. In diet you are taking the copper. Various number of dietary sources are there. Okay. So you are taking in the diet. What happened in diet? It is absorbed in the small intestine. Okay. Intestine. Diet you are taking and through the process of absorption it is absorbed in the small intestine. Next, what will happen? Then there are the role of two transporter protein. That one is ATP7A and another is your ATP7B. Okay. So there is a transporter protein that is of your ATP7A. And what is the role of this ATP7A? This will help in the intestinal absorption of your copper. Alright. What is the role of this ATP7A transporter? It is a transporter protein. Okay, it is a transporter protein. So the role of ATP7A is that it helps in the intestinal absorption. And second thing, it helps the transportation of this copper into the where? From small intestine, this copper enters into your liver. Okay. So what this ATP7A transporter will do? It helps, first thing, it helps in the intestinal absorption and helps to transport this copper from small intestine to liver. An important thing is that the copper which is traveled from small intestine to liver, it is bound with your albumin. Okay. So copper albumin complex will be formed, and in this complex will travel to the liver. And when this enter into the liver, then what happened in the hepatocytes? This albumin will release out, and then copper remaining in your liver. Okay. Next, we will understand what happened in the liver. When copper enters into your liver, then what is the fate of that copper further? So this is your liver. Okay. In liver, what happened? Then there is a role of another transporter protein, that is your ATP7B. Okay. So what is the role of this ATP7B? This transporter protein, first thing, helps in the excretion of copper. What it will do? Two things it will do. It helps in excretion of copper into your bile. Okay. It helps in the excretion of copper into bile. And what is the second function? Second function is it helps in the incorporation of copper with apoceruloplasmin, and copper and apoceruloplasmin will combine together to form ceruloplasmin. And in this ceruloplasmin form, copper is transported to the whole body. Okay. So once again, I am telling you what happened. First thing, you are just taking any dietary sources of copper. You are just taking the copper in any diet. Okay. Then what will happen? That copper enter into your small intestine. And then in small intestine, there is a transporter protein, that is your ATP7A. And that transporter protein, that is ATP7A, help in the intestinal absorption of your copper. And second thing, what that transporter protein do? That ATP7A will transport the copper from small intestine to liver. Now what happened? Now your copper enter into your liver. Okay. What happened in the liver? There is another transport protein, that is your ATP7B. And what is the role of this ATP7B? It played two important roles. First thing, it helped in the excretion of copper into your bile. Okay, from liver through bile, copper is excreted. Second thing, this ATP7B helps in the incorporation of copper in the form of ceruloplasmin. So what happened? First, copper combined with your apoceruloplasmin, and with the help of ATP7B, it is incorporated and it will form your ceruloplasmin. And what is ceruloplasmin? It is the transporter protein. In ceruloplasmin form, copper is transported into whole body. Okay, so this is the normal mechanism. Now, what happened in this Willson disease? In Willson disease, this ATP7B gene is defective. If your ATP7B gene is defective, then of course this transporter protein is also not there, because that gene is defective. So there is no ATP7B transporter protein. Okay, this protein is not there. If this protein is not there, then these two functions will be performed. No. It's not possible. Okay. Then there is no excretion of copper into bile and no incorporation of copper into ceruloplasmin. Isn't it? Because this protein is not there. Okay. So what happened in Willson disease? What are the clinical features of Willson disease? Okay. Now you understand what is the role of ATP7B, and if ATP7B gene is mutated, then what will happen? If this ATP7 gene is mutated, then two functions of what happened? Two functions of your ATP7B is affected. Okay. What are two functions? That is excretion of copper into bile, and second, your incorporation of copper into ceruloplasmin. Alright. Then what are the clinical features that the patient will present? So this disease is also known by another name, that is hepatolenticular degeneration. Okay, so another name of your Willson disease is hepatolenticular degeneration. Okay, you should know the another name. Why it is known as hepatolenticular? Because the copper is deposited into, hepato means what? Liver. Okay, so the copper get deposited into your liver, and copper get deposited also into your lenticular nucleus of brain. Okay, in brain there is a basal ganglia. Okay, you know the anatomy. In brain there is a basal ganglia, and the basal ganglia of brain there is a lenticular nucleus. Okay. So the copper get deposited in two organs. One is in liver, another is in the lenticular nucleus of brain. Alright. That's why the disease is also known as hepatolenticular degeneration. Alright. So now what happened? What are the clinical features? Then if copper is not incorporated in the form of ceruloplasmin and copper is not excreted into bile, then what will happen? There is an excess form of copper. Because it's not going outside the body into the bile and it is not incorporated, then excess bile get deposited into your liver. Okay, sorry, excess copper will get deposited into your liver. So when excess copper get deposited into your liver, then what it will form? It will call hepatomegaly. Understand? So why hepatomegaly is there? Because excess copper get deposited into liver. Second thing, now there is the excess copper in whole body. Okay, that excess copper is first deposited into your liver, results in hepatomegaly, and that excess copper get deposited into where? That excess copper get deposited into your this lenticular nucleus of brain. Excess copper, what will happen? Excess copper deposited where? Is deposited into lenticular nucleus of brain. And because of deposition of this excess copper in lenticular nucleus of brain, patients present with what? Neurological symptoms. Okay. Neurological symptoms. So hepatomegaly is there, neurological symptoms will be there. What kind of neurological symptoms? Like patient having a tremors, behavioral changes, mental retardation are there. Okay. So these are the neurological symptoms comes in the patients of your Willson disease. What other things? What other clinical findings will be there in Willson disease? So another finding is that of these copper. The excess copper, what will happen? The excess copper get deposited into the eye. Okay. Next, where is the deposition? In eye. What are the changes in the eye? The excess copper get deposited in where? It is deposited in Descemet's membrane. It gets Descemet's membrane of cornea. Descemet's membrane of cornea. So excess copper get deposited into the Descemet's membrane of cornea and it will form a golden brown ring. It will form a golden brown ring in cornea and that ring is known as what? That ring is known as KF ring. KF ring. Okay, what is the full form of KF ring? Kesher-Fisher ring. Kesher-Fisher. Okay. So the question is asked, very important star point. In which disease KF ring or Kesher-Fisher ring is deposited in cornea? The disease is your Willson disease. Okay. Why? Because there is an excess copper and that excess copper get deposited into the Descemet's membrane. In the cornea there is a membrane-like structure. Okay. And that excess copper get deposited into the your Descemet's membrane of cornea and it will form a golden brown ring. Like that. Okay. Form a cornea, there is a golden brown ring is there and that ring is known as KF ring or Kesher-Fisher ring. Okay. What another feature is found in eye in Willson is there is a sunflower cataract. Okay. The lens of eye is get opaque. And what kind of cataract is there? You remember sunflower cataract. Okay. So these two things you must remember for MCQ as well as your Viva. That where is KF ring is present in which disorder? That is Willson disease. And what kind of cataract is found in Willson disease? So there is a sunflower cataract is found in the Willson disease. Alright. Okay. So these are the clinical features of your Willson disease. Now comes on the diagnosis. How you will diagnose the disorder. Okay. How you will make the diagnosis of your Willson disease. So what is the diagnosis? How you will make diagnosis? So first thing is there, if ATP7B gene is defective, then is it copper incorporated into celluloplasmin? No. Then what about the level of celluloplasmin level? Low or high? Nabil, what is the level of celluloplasmin level? Low or high? Low. It will be low because it cannot deposited in the. It's not deposition is not there, na? Because ATP7B gene is not there. And what is the function of ATP7B gene? So ATP7B transporter protein helps in the incorporation of copper into celluloplasmin. So if ATP7B transporter is not there, will celluloplasmin will form? No. Okay. Then if celluloplasmin is not there, the level will be low or high? Of course low. Okay. Got it? So first what you will do? You will check the celluloplasmin level, serum celluloplasmin level. Okay. So serum celluloplasmin level is low. Second, you will check the serum copper level. Okay. Then what about serum copper level? This serum copper level. Remember, if you will check the serum level of copper, then it will be low because that serum is get deposited in all your organs. So when you will withdraw the blood and you will check the serum level, then serum level will be low. Okay, don't confuse it. And third is about urinary level. If you will check urinary copper level, then what about urinary copper level? It will be high because your excess copper get excreted out through the kidney. Okay, that's why remember your celluloplasmin level is low, your serum copper level is low, but your urinary copper level is high, and it is about more than 100 microgram per 24 hours. Got it? Okay. Next is what is the question asked? What is the gold standard? Which is the gold standard diagnosis for your Wilson? So gold standard is that of your liver biopsy. Okay, liver biopsy. Okay, so if the question is asked which is the gold standard test for the diagnosis of Wilson, then you will do liver biopsy. You will do the biopsy of liver and you will check the liver. Okay, alright. So by this you can easily make the diagnosis. And second and last thing, you will do the examination. Okay. Lastly, what you will do? You will do the examination. Okay. Examination, what finding will be there? There is a finding of KF ring around the cornea. Pressure-Fisher ring is there on examination. You will see the patient present with the neurological symptoms. Okay. Behavioral changes, mental retardation, tingling sensation was there. Okay. And if it's an advanced state, then sunflower cataract is there. Okay, so by this you will easily diagnose the Wilson disease. Now comes on the treatment. How you will treat the Wilson patient? What treatment you will give to Wilson patient? Okay. So what is there? There is a excess copper, and copper excess that excess copper get deposited into your all of your organs. So what is your main treatment strategy? Main treatment strategy is what? You will remove that copper that get deposited everywhere. Okay. So first thing what you will do? First thing, dietary restriction. You will do diet restricted in copper. Okay. So what you will do? You will advise patient to copper restricted diet because already there is a copper excess. So that's why there is excess of copper. That's why that excess copper get deposited everywhere in liver, in your brains and your eyes, etc. Okay. So what thing you will advise first? You will advise patient that you should take a copper restricted diet. Like you should not eat so much of your like chocolates. Chocolates are rich sources, nuts, like nuts, your kidney, liver. Okay. So these etc. So these are the sources of copper, and you will advise your patient not to eat all these things. Alright. Second, what you will do? Second treatment is your chelation therapy. Chelation therapy. What you will do in this chelation therapy? You will give the chelating agents. Okay. Excess copper is there, so you will give the chelating agent. Like, what kind of chelating agent you will give? Penicillamine, yes. Like your penicillamine, okay? Penicillamine. And you can also give BAL, that is British anti-Lewisite. BAL is what? British anti-Lewisite. That's also act as a chelating agent. Okay, so you will chelate that excess copper which is get deposited in the body. Third thing, what you will do? You will give zinc. Okay, zinc you will give in for the treatment of Wilson because it increase the absorption of copper. Okay, what is the role of zinc in Wilson? Because it increase the absorption of your copper. Alright, it increase the absorption of your copper. And lastly, if patient is not responding all this medical therapy and all this therapy, last thing what you will do? You will do liver transplantation. Alright, so what are the treatment strategy you will do in the patient of Willson disease? First, as there is already copper excess in the body, so you will advise first patient not to take so much of diet which is rich in copper. So you will advise your patient just take copper restricted diet. So what you will write? Advise the patient not to eat so much of chocolate, not to eat so much of nuts, kidney, liver, etc. Second thing, what you will do? Second, there is a excess copper in the body. So what you will do? You will do the chelating, through the chelating agent, you will do the chelation therapy. And most common chelating agent which is used in Wilson is that of penicillamine and your British anti-Lewisite. This British anti-Lewisite, do you remember? We have studied it before. What is? This is your inhibitor of your third complex of ATC. Do you remember? So it is a complex 3 inhibitor of ATC. Okay. Anyway, penicillamine, and after that, another treatment what you will perform? You will give zinc. What zinc? What is the role of zinc in treating Willson disease? Zinc will increase the absorption of copper. So when copper absorption is increases, it will remove out through the kidney outside your body. Okay. Next, if patient is not responding all these things, the final treatment is your liver transplantation. Alright, so this is still here. This is all about the Willson disease. Next question I am telling for the PG students especially. One question is asked in this question. What is the role of zinc in Willson disease? Okay. How the zinc is useful in treating the Willson disease? What is the role? How it will increase the absorption of your copper? This question is for PGs. The question is that how zinc is used in Willson treatment. Okay, Willson treatment. Okay, this question is for especially for the PG students, not for UG. UG up to that is sufficient. Okay, but for PG students, additional question is that why zinc is used in Willson disease? What is the role of zinc? So zinc what will do? This is your intestine. Okay, so this is your intestine, and you give the zinc to the patient. Okay, you just give the zinc to the patient. Okay? What is the role of this zinc? This zinc will stimulate. This zinc will stimulate the formation of one protein. Okay, when you will give zinc, then what is the role of zinc? Then zinc will stimulate the formation of one protein, that is of your metallothionein. Okay, remember the name. Then when you will give the zinc, then what is the role of zinc? This zinc will stimulate the formation of this metallothionein protein. Okay, and what is the role of this metallothionein protein? It will increase copper absorption. Okay, what is the role? It will attract all the copper, and it will increase all copper get attached with this metallothionein, and it will increase this copper absorption. And when this copper is increased, and then it will finally excrete out through the urine. Okay. So one thing you have to remember is that some additional question is also added for the PG students. Okay, what is the role of zinc and what is the mechanism of action of zinc in Willson patients? So mechanism of action of zinc is that it stimulates the formation of this metallothionein protein where? In the intestine. Okay, in the intestine this metallothionein protein formation is there, and what is the role of this metallothionein protein? It will bind more copper. Okay, so it will bind with more of the copper in the body, so it will increase the absorption of copper, and when that ultimately excrete out through the urine. Alright, so this is all about your Willson disease, and this is a must read topic for short note. Okay, you have to prepare like this. It is an important topic for both UG and PG. Okay, so thank you all.